All right. Hey, everyone. Today we are with Alex Yu. He is currently an instructor at the Cedars-Sinai Medical Center and an incoming assistant professor at the Bioengineering Department at the University of Maryland. He is also the associate director of the Spatial Molecular Profiling Core Facility at Cedars-Sinai, and his research focuses on studying the spatial biology of the tumor immune microenvironment in order to propose novel biomarkers to predict clinical outcomes and novel therapies to improve clinical outcomes. Today, he's going to present his work on spatial biology of several cancer systems using highly multiplex spatially resolved measurements of proteins and RNA, and he's going to discuss his efforts to construct spatial atlases of both solid and hematological malignancies and how to translate spatial data into biomarkers and novel therapeutics. With that, throw it over to Alex.
All right, thank you very much, Hector, and thanks very much for the organizers for this opportunity. Uh, yeah, as Hector mentioned, I'm currently an instructor at Cedars-Sinai, and a lot of the work that's done at Cedars-Sinai is done through our core, spatial molecular profiling core. So I'm very grateful for the core as well, right? It gives us a lot of opportunities to collaborate across different disease systems and different tissue bank resources. So hopefully over the course of the talk, I'll be able to touch on those and sort of build out this story. So I just want to start with a little bit on the field. There's a lot that's happening right now in spatial biology. I've kind of split it up here in terms of methods and applications. Of course, on the method side, we're always coming up with new ways to measure more more analytes, more say biological features, right? Whether it's epigenetics or post-translational modifications. Of course, our very own Ahmed is really pushing in this field with his work on metabolites. On the other side of methods, we have applications, and this is really where I want to spend my time today. So I've listed three applications here. The first is atlases. This is sort of the lowest hanging fruit of grabbing as much tissue as possible and running it on these machines. Uh, we also have predictions, so predictions and prognostics. On this side of things, we're interested in grabbing as much tissue as we can, learning what that tissue looks like, so that when a new piece of tissue comes along, we can say something about that new piece of tissue. And then finally, we have therapeutics. Therapeutics is a bit forward thinking. I don't think we're there yet, but the idea here is, of course, to look at enough tissue to be able to predict what will happen when we perturb that tissue in a particular way, right? So I don't think we're there yet. I'll touch upon this a little bit later, but what I really want to focus on today is this sort of second application in terms of predictions, right? Here I'm just calling it biomarkers for prognostics and prevention. So a little bit on this topic. My background is engineering and material science, so I was never really exposed too much to the clinical side of things. Now being at the hospital for a couple of years, started to learn a little bit more about it. It's of course a very complicated process, a lot's at stake. So when new patients come in, uh, it's the imperative of the hospital to make sure that we're learning as much about these patients, but also not doing them harm and not subjecting them to unnecessary testing. So, for example, this is a flow chart based on prostate cancer, and it looks a little bit flippant or silly with this little guy at the bottom left, but it's quite serious, right? It's a series of tests that help us to determine how serious the disease is. So through a clinical decision-making tree like this, yeah, we're trying to answer maybe two main questions, right? The first is, what is this patient's diagnosis, right? How much risk do we assign to this particular case? And then two, can we learn something about the molecular basis for the disease? Of course, these are both really complex questions. And so the hope here is that we can come up with concise measurements, simple measurements that don't subject the patients to too much hassle. They're not that invasive, that can capture these two questions. And sometimes these are empirical, right? A larger tumor leads to X, Y, or Z. Sometimes they're more hypothesis driven. There's a particular oncogene that could suggest a particular treatment. But in the end, the hope is for these tests to be sort of as minimal but as incisive as possible, right? So I'm going to be calling these biomarkers. So On the topic of biomarkers, I'll just start with a simple analogy. This is probably one that a lot of people have heard by now, but the analogy is this: given a bowl of fruit, do I want to eat this fruit? Right? And there's a lot of reasons not to eat the fruit. Maybe you're sort of washing the fruit and you see a little lump on the grape. What do we do? So you might ask a friend who, let's say, went to fruit school and fruit residency and fruit fellowship, should I eat this fruit? Maybe you take a survey of 5,000 people who ate this fruit. And if 80% of them die within 5 years, that's another good reason not to eat this fruit. But the point is that we need to learn more about the fruit. We need to measure it some, right? To extend the analogy, there's a lot of different ways we eat fruit. You can make a smoothie. Of course, this is a little bit tricky to figure out if the fruit is good or bad, right? Everything is blended together. If you have a bowl of fruit, it's a little bit better. You can say identify a moldy grape, but I mean, it's a bowl. The grape has sort of been rattling around. You don't know what's touched what. With a fruit cake, though, I think we're really getting somewhere, right? There's a moldy part of the fruitcake, maybe I can cut that part out and still eat the rest. If you show me what 10 good fruitcakes look like and you show me an 11th one that doesn't look the same, I might not want to eat that 11th one. Right? So I don't think I'm being too mysterious about the analogy. The fruit is, tissue, cancer tissue, and the way we consume these fruits is the different ways we measure it, whether it's through bulk analysis, single cell suspension methods, or spatial methods. So I really can't take credit for this analogy. It's been around for a while. But what I'd like to push on is what I'm just calling the sort of next stage of this analogy, or the evolution of the analogy. And it's this gooey cake. So this is a pineapple upside down cake. I'll have to admit that, culturally, I don't really eat a lot of these, but, I can still appreciate a delicious looking cake. You've got pineapples, that's good. Cherries are good. Cake is good. So, uh, I think what makes these cakes really special, though, and you kind of see it listening here, is a caramelization, right? That sort of gooey flavor spread that is going to come out from the fruit, right? And whether you prefer cakey cherries or more pineapple cherries, they, it, the flavor comes from how the fruits are interacting. So if there's anything you take away from this talk, I hope it's to start, framing your spatial thinking in sort of more interesting and interactive ways. So this is the analogy, but to move on to the biology, the first topic I wanted to talk about is ovarian cancer. So just to introduce the topic a little bit, ovarian cancer is a leading cause of cancer death in women. So despite it being sort of outside the top 10 in terms of prevalence, it's number 5 in mortality. And the reason for this is that it's diagnosed very late. So, of course, what that means is that the tumor has a lot of time to sort of grow and co-evolve with its environment before it sees any treatment. Now, in terms of the tumor itself, uh, there's these sort of 3 main cell categories that we typically talk about in tumors, right? We've got tumor cells, stromal cells, and immune cells. And, one way to think about this is you have this sort of stromal bed of tissue. You've got your tumor cells, and you have immune cells that are maybe interspersed throughout. In reality, though, the story is more complex. So this is a piece of tissue. Of course, a pathologist can look at this and tell you instantly what's there. I went ahead and cheated and asked the pathologist, but just to draw your attention to a couple things, on the left side, we have a bunch of immune cells. So these are, generally smaller, and they have these darker nuclei. On the bottom right, I'm just highlighting an inset area where there's both tumor cells and fibroblasts. So the tumor cells are a little bit darker, the nuclei are much larger. And, of course, all these cells have different shapes and sizes. This is H&E, so we're not learning too much about specific molecular features. And that, of course, is what we're looking for with sort of modern spatial omics, right? The actual molecular features that characterize these cell behaviors. So for a long time, as we've tried to describe the spatial composition of these tissues, we've often there's been both quantitative and qualitative metrics, right? So in this case, in this study, these authors tried to describe all these different components: the immune infiltrate, how many CD3 T cells are present. There's a stromal scoring metric, which is more qualitative or semi-quantitative, perhaps. And then there's tumor cell composition, so that can be of course, there's not very much resolution here, right? We know that there's a lot of different cell types that are present, a lot of different interactions that are really not being represented with, say 3 metrics like these. And the problem for that historically is that most of our clinical descriptors are just semi-quantitative at best, right? And that's not because people don't want to learn more, but it's been a limitation of technology. So for a long time, images like these are sort of the workhorse methods. There's not that many channels we can look at, say, and you may not necessarily be able to trust the quantitation, the intensity of these signals. So what we're left with are these semi-quantitative metrics where you might take a look at these 3 tissues and say that the one on the left is infiltrated, partially infiltrated in the middle, and then not infiltrated on the right. And so, this is not very satisfying to me as an engineer, of course, but especially when it comes to thinking about how we define these thresholds, right? And how rigorous we can be about this. And what's the difference between the lowest infiltrated tissue and the highest partially infiltrated tissue? Nevertheless, there's still a lot we can learn with these types of analyses. So I'll just highlight one particular study that I found interesting. And I'm trying to communicate the idea that, spatial information, it's an evolution of sort of simpler methods, right? So for example, in this study, these authors took a look at a large cohort of ovarian cancer patients. First, they just noticed that in terms of platinum resistivity, right? This is sort of a clinical observation. Of course, patients that respond to platinum often have better survival than patients that don't. Now, when you just look at these 2 categories, that the patients that respond to platinum, where they had less stroma, right? And then the patients that were platinum resistant had higher stroma. So this is interesting. This is sort of a bulk-ish measurement, right? It's the number of fibroblasts in the image. It's not a very complex measurement. When we just look at the stroma, so if we sort of stratify these patients by high or low stroma, we again see that there's a survival difference, right? But so how do we contrast this with the previous observation? Well, the first observation was a clinical observation, and this one is a sort of morphological observation, right? We're just looking at the tissue. We're kind of blinded to the platinum sensitivity. And then finally, the last piece of this figure for this study was they looked at what they're calling CD8 stroma infiltration. So looking at T cells that are at least 1 cell away from the tumor, they call that stromal infiltrating. And again, they see that low CD8 infiltration in the stroma is correlated with low survival. Right, so the progression here is kind of going from a clinical observation to a pure morphological observation to now a marginally more complicated morphological observation. In this case, CD8 infiltration to stroma. But you can imagine this one piece of information actually contains information about T cells, fibroblasts, and tumor cells, right? Because you have to be away from the tumor and into the stroma for the T cell. So this is kind of how we go from, bulk measurements, clinical measurements, all the way to spatial. Of course, we'd like to go further on the spatial. So to talk about that, I'll be discussing a particular cohort that we put together with our collaborators at UCLA. She was actually here at Cedars when the cohort was assembled. But anyways, we're looking at 42 patients. So these are ovarian cancer patients. They were, as I mentioned before, this cancer is detected late. So all these patients at the time of diagnosis had both primary tumors and metastatic tumors. So we collected these tumors from these patients. Luckily for ovarian cancer, surgery is part of the standard of care. So we collected the tumors from these patients. The patients then received chemotherapy. Now, in this particular cohort, every single one of these patients had a recurrent tumor. So when the tumor recurred, we again collected the tumor. These tumors, of course, now experience platinum-based chemotherapy. And using this tissue, we collect representative regions from each tumor, up to 3 regions, and we assemble them on a tissue microarray. And that's what we'll be analyzing today. So the time to relapse is actually the critical clinical variable here. In this case, some patients relapse very early, then just a few months. Some of them survived for upwards of 4 or 5 years. And there's a sort of a natural separation around 15 months. So we considered patients that relapsed before 15 months to be early relapsers, and those that relapse later to be late relapsers. So that's kind of the main sort of clinical anchor for the study. Now, in particular, working with different cancer systems, especially as a core facility, our collaborators, the biologists, are really the experts on their samples. So there's quite a bit of curation that goes into all of these data sets. Um, Once we assemble the tissue microarray, we use a technology called imaging mass cytometry. So this technology allows us to measure 40 proteins at about 1 micron resolution. I don't think I really need to go into details for this audience, but very briefly, we take the tissue, we slap it on a glass slide, and we stain it with antibodies that have been conjugated to metal ions. Those ions are then quantified pixel by pixel in the machine. The ions, of course, allow us to do multiplexing without worrying so much about spectral overlapping with fluorophores. And then our job is to, kind of compress this 40 or so plexed image and interpret, which cells are present and which proteins are expressed in those cells. So at this point, we kind of go back to the collaborators, we have a conversation with them, try to figure out how we can interpret these images. Specifically, we try to remove various technical artifacts or biological artifacts that might emerge, for instance, necrotic regions or places where the tissue has folded on itself. And the result in this particular study is about a 30-plex protein analysis of markers that are pertaining to different stromal types, whether it's smooth muscle actin or tumor cells, just pankeratin, so on and so forth. And there's a lot of spatial data that's out there now. A lot of spatial data is sort of maybe more beautiful than this one, but this is ours. So when we take a look at these images and we start interpreting it, of course, we get images like this one on the left. What I'm doing here is sort of a very, very simple analysis to start, right? Just focused on these three main categories of cells, whether it's immune cells, fibroblasts, or the epithelial cells, which are the tumor. And we'd love for some significant correlations to emerge, right? That if there's some kind of silver bullet predictor of whether these patients will relapse early or late. So to start with, we just kind of compare the cell type compositions of these tissues. And what we find is that at least at the primary metastatic and recurrent level, there's really no difference, no significant statistical difference between the tissue composition. So that's kind of read out. Actually, let me get my laser pointer here. But it's sort of read out by this ternary plot. And I won't go into detail, but what this plot is showing that there is a little bit more immune cells. We observe more in recurrent tissue, but it's not a significant difference. Looking ahead to the relapse state, again, there's no significant difference between patients with early relapse or late relapse. There's no difference in their cell composition. There's maybe a little bit, we've observed less tumor in the early relapse, which is a little bit counterintuitive, but the difference isn't significant. The one significant difference is that we have several different metastatic sites, right? Ovarian cancer primarily metastasized to the omentum, which is sort of the sort of nearby tissue. But we also often find it in lymph nodes as well as other tissues. In the lymph nodes, we see, as you might expect, we see quite a few more immune cells. But of course, whether that's actually a function of the lymph node having more, the tissue itself having more immune infiltration, or if it's just a function of the sort of the tumor site and where the tumor is invaded into, we don't really know. So that's kind of a question that needs more context that I'll be presenting in a little bit. So this is really the only significant and sort of, frankly, trivial observation. And at this point, it's kind of disappointing, right? We really hope that there would be a difference, but there just isn't. And I think this really makes sense. When we think of back to these pineapple upside down cakes, right? There's a million ways you can make a cake. And, just because you add a little bit more pineapple, it's still a cake. It's still a pineapple upside down cake. It doesn't necessarily mean it'll taste better or worse. So, given this sort of lack of silver bullet solution to describing these tumors, well, what can we do? So along these lines, I'll just say that, there's a lot of cakes here, but there's something about these top three cakes that I'm presenting that makes me not want to eat them, right? They're just lacking a little bit especially compared to the one on the bottom, right? Like that's a beautiful cake. I really like this one at the bottom here. So obviously what we're kind of, I'm kind of articulating, but I think everybody kind of understands is that it's the spatial composition of these tumors that we really need to think as the next level, right? Beyond just quantitating the number of cells, it's about how they're arranged. So along those lines, I'll just talk about how we start thinking about this and move forward. So I'm showing you here on the left another particular tumor sample. It's interesting because there's this sort of swath of tumor cells on the top, and the rest is other cell types. But when we look at these categories, we could see that the clearly the tumor cells are segregating from the other cell types, and so to quantify that, we use a method called G-cross. So I won't get into too much detail, but essentially we're looking at how different cells interact, whether it's how they interact with themselves or with other cell types. So in this case, again, a simple measurement, we're just looking at stroma, tumor, and immune cells. And in this particular example, we could say that, say, the tumor interacts very strongly with itself, and tumor interacts very, very weakly with, fibroblasts. So this particular image with that sort of set of spatial interactions would fall under this particular cluster here at the top. And what I'm showing here are 9 clusters that we've generated using these different G-cross metrics. So for example, the top one was a sort of strong tumor-tumor interaction, weak fibroblast-tumor interaction. Here's another category on the bottom here, this middle category where again the tumor and the fibroblasts are segregated, but now we have immune cells and fibroblasts interacting more tightly, right? So that ends up in a different category, and you can kind of see how that vibe is different. And on the bottom here, we've got what I'm calling a well-mixed periodic tumor, right? And in this case, all the different cell types are interacting fairly well, and we have this sort of more dispersed, diffuse tumor type. So along the way, so these are basically sort of 9 spatial phenotypes. Along the way, we also look at sort of phenotypes, sample level phenotypes, let's say, that are generating using just the cell proportions. And when we kind of map these against each other, with the here on the x-axis, we have the different spatial sample types. On the y-axis, we have different cell proportion based sample types. And you can see that there's not a one-to-one correlation, right? For example, for this immune high cell proportion phenotype, there's a lot of different ways you can generate that spatial, right? So, you can stare at these plots all day, but I'll just cut to the chase and say again, there's no significant association with our clinical endpoint, right? There's no spatial orientation, there's no spatial phenotype, there's no cell proportion phenotype that's significantly correlated with early relapse. So at this point, it's kind of disappointing, really. Uh, why isn't it working, right? Why aren't we getting why aren't we learning anything about how to predict early relapse? So I'll just talk about this for a little bit. As I mentioned before, this tissue microarray has been curated. So it's done in conjunction with a pathologist who kind of looks at the tissue and identifies the sort of most relevant regions, right? And so this is kind of the first sticking point to why it's difficult to come up with these biomarkers, right? Because of course, when we're punching out regions of tissue, we're losing some information. There's always this risk that there's other predictive features that are elsewhere in the tissue that could be used to make a prediction. But the catch-22, the other side of this is, this strategy allows us to profile 42 patients worth of tissue with triplicate tissue across 3 tumor sites, right? If we were to do that with the same technology at 42 patient scales, you'd blow through $1 million, $2 million. It's just not practical, right? On the other hand, if you're going to do whole slide imaging, which is another viable strategy, uh, how many patients can you really look at, right? You're not going to be able to say anything statistically interesting. So that's the first challenge with these biomarkers. The second is maybe more related to the actual sort of physical construction of the tissue microarray, right? So I'm trying to illustrate this here on the bottom. We have a particular sample that has a lymphoid aggregate on the left side, and we have these sort of tumor clusters, these sort of swirls of tumor cells on the bottom right. And so a pathologist has, we punch out this tissue, pathologist kind of points out, like, I think you should look at this area. And we then punch out that particular core and assemble it on the microarray. But the microarrays are a hot commodity, so a lot of people are working on these microarrays. And as we go, we might be looking at a piece of tissue that's quite a bit removed from that first representative slide, right? We're talking, hundreds of microns, even millimeters apart. So you can imagine what happens with this tissue is that it changes in the Z dimension. So in this case, you might expect, say, the tissue to slide a little bit to the left. And when we're looking at the same expected area, maybe you lose that lymphoid aggregate and you're sort of looking at only the tumor clusters now. And so that's actually exactly what happened with this particular image. Here I'm showing you the processed image for this same sample. And you can see that indeed that lymphoid aggregate that we had on the left, that's shifted out of the picture. And now we just have these sort of tumor swirls. Right? So this is the second problem with this general strategy, is that what used to be a represented region has now maybe, let's say, become semi-represented, right? So I think that the main challenge here, right, is to think about uh, what kind of information we're gathering from the tissue and its local context. So along those lines, I'll just talk about a strategy that we use to start to tackle this problem. So what we did is to develop a sort of spatial enrichment strategy. The first step here is to try and develop a spatial density metrics. So a kind of scoring for each cell that describes its local microenvironment. As an example here, I'm showing you yet another H&E slide. You can see that there's sort of a couple main regions of tumors. We have these sort of blobs, these islands of tumors, and they're surrounding here by these very, very small cells, which are immune cells. Here's the IMC image. So hopefully you could trust that it is the same image, but again, it's removed by several, I believe a millimeter almost. And you can see that the tumor swirls, the tumor islands have kind of changed their shape, right, in the Z dimension, but they're sort of still there in the same general areas. So right now we have this challenge I talked about earlier, where this is likely a lymph node metastasis. It is when we just measure the amount of immune cells here, we're just flooded with immune cells. But is that a function of the tumor, or is that just the tissue that it happened to metastasize to? So in this case, what we'd like to do is kind of figure out where the tumor is and curate the tissue further based on that analysis. So one thing we can do, right, is we can try and look for the cell type specific density, in this case, the tumor specific density of every cell in this image. So we perform this calculation, and we can see one thing we've added to the calculation is interaction radius. The idea here is that if we're two cells in communication, but you're so far away that I can't touch you, I can't hear you, I can't smell you, right? Like biologically, molecularly, I can't sense you, then you're sort of irrelevant to me, right? So spatially, we introduce this interaction radius to account for this. And what that results in is a sort of set of tumor enrichment scores, where we're able to score each cell on how likely it is to interact with tumor cells. Finally, the last thing we can do, and it's relatively simple, is just to apply this threshold, right? So as long as we extract the cells that have this particular tumor enrichment score, we can get a sense for which of these cells are actually sort of influencing the tumor and which of them are maybe doing their own thing. So with these scores, we developed a couple strategies. The first thing, it was to just look at lymphoid aggregates, right? So the most well-known is a tertiary lymphoid structure in a sort of tumor context. And using these spatial enrichment scores, we're able to isolate these lymphoid aggregates. We apply a connectivity filter as well. And this is actually already a highly observed phenomenon. TLSs are generally associated with good outcomes, and we also observe that our early relapse patients have fewer TLS in their tissue. Another thing we did is try to provide more context-specific niches. So we're calling this a digital biopsy, but the idea here again is just like I said, right? To really focus on particular tissue compartments with a quantitative eye. So in this case, what I'm trying to show you are just two different strategies where we're curious about whether or not, cells that interact with any tumor cells have a particular phenotype, particular spatial arrangement. And we'd like to compare that to cells that are really sort of in these tumor nests, right, that are associated with high concentrations of tumor cells. So we applied these two digital biopsy strategies, one looking at just tumor contact, one looking at actual tumor enrichment. And when we start looking at these spatially biopsied regions, we start to see more significant statistical interactions, right? So again, on the left side, on this ternary triangle plot, we didn't see any significant interactions, but when we start focusing on different spatial compartments, we some interactions start emerging, especially around the fibroblast compartment, interestingly enough. So it's really the stromal regions that sort of had the most say in making predictions. We can also then look at the spatial temporal associations, right? We have tissue taken at primary metastasis and recurrence. And what we find is that it's really the difference between the primary and metastatic or primary and recurrent tumors that's the most predictive, not so much between metastatic and recurrent tumors. So that's really what I want to focus on. The study itself also contained a number of follow-up follow-up analyses, especially using other omics types to explore a particular change in particular B cells and plasma cells. So we combined this analysis with Visium gene expression data and sort of targeted multiplex immunofluorescence to explore how B cells segregate and how that influences early relapse. So I won't get into too much detail. Of course, if you're interested in this particular topic, please check out this paper as recently published. But in conclusion, this study was really powered by these 42 patients where we had matched samples over the course of tumor progression, right? And I kind of talked about how we went from this sort of historical space of qualitative eye tests and semi-quantitative measurements to trying to quantitatively and rigorously define these different spatial compartments and make useful comparisons, right? The whole point is to try and make these apples to apples comparisons between relevant pieces of different patient samples. And so we did that through this sort of spatial method of digitally isolating specific regions. Now, there's a lot of other content in this paper that I will skip ahead of, well, I'll skip over for now. But of course, we talked about other predictors of early relapse. We did a sort of sub-study on the concordance between IMC and H&E. And we developed this sort of unifying hypothesis for how B cells and plasma cells in particular help predict early relapse in ovarian cancer. But I did want to take the rest of my time to kind of shift gears and talk about another system. First, there's most of what I just talked about is still focused on spatial orientations and arrangements, right? But I kind of hinted at this like that. I don't think that's really the whole picture, right? So kind of moving back to the cakes, most of what I talked about is sort of related to this idea of spatial niches, right? So in this case, we have this sort of pineapple and cherry niche in this cake. Sometimes it's shaped like this circle or ellipse or maybe a semicircle. I don't know what's happening with this cake. This is an insane cake. But what I propose maybe is that even if this cake looks crazy, we shouldn't prejudge it, right? And there's even if the shapes are kind of strange, that doesn't capture the whole story of the cake to me. So I think what we really need to focus on again is that molecular detail that we have. So for this topic, I'll move on to a different cancer system, Hodgkin's lymphoma. So Hodgkin's lymphoma is unique among B-cell-derived lymphomas in that the tumor itself is actually quite rare, right? The tumor cells are called Hodgkin's Green-Surberg or HRS cells, and they comprise usually less than 10% of tissue. Most of the rest of the tissue is immune cells. So we have this sort of playground of immune tumor interactions. To the topic of biomarkers, there are a number of useful biomarkers in Hodgkin's. For example, this is a study from our collaborator at BC Cancer in 2010, where they found that macrophages were a useful predictor of patient outcomes, right? The more macrophages you have, the worse off you might be. So this is sort of, again, the thesis for Hodgkin's and many other diseases, which is that the way that immune cells interact with the tumor, not only can we use that to predict patient outcomes, but this is going to be a really important interaction, right? This is what we're going to modulate for immunotherapies and hopefully curing this cancer. So just to focus on the biomarker a little bit, the way that it works is, again, we take this sort of simple image of the tissue. In this case, it's just CD68 positive staining. There's no spatial information involved here, but we just stratify the patients by sort of high, medium, or low CD68 percentage. And just by doing this, we can see differences in survival. So of course, what we'd like to do and what we attempt to do in this study is to advance understanding, right? To push forward with spatial data. To start with discussing the cohort itself, we have 92 patients, 95 patients, 73 of which relapsed and after chemotherapy, and 22 of which were cured by chemotherapy. So in comparing these two populations, we're interested in chemosensitivity of these patients, right? Are there spatial features that help predict response to chemotherapy? Within the relapsed and refractory patients, we've got 26 early relapsers defined as less than a year and 47 late relapsers. By comparing these two populations, we're interested in sort of immune spatial mechanisms of chemo resistance, right? I'll also offer that among the late relapsers, we're sometimes talking about 20 plus years, 20 plus year gap. So thinking about that, it's not just a matter of whether this tumor has been slowly growing over the course of 20 years, right? That's almost certainly not the case. Likely it entered some kind of dormant or senescent state and reemerged perhaps after the loss of some control mechanism, right? So again, we're interested in immune mechanisms of control that are different between late and early relapsers. And finally, we have for all these relapse patients, we have both diagnostic and relapse samples. So in this case, we're kind of curious about how the tissue is changed, how it's remodeled after experiencing chemotherapy. So this particular image, this is a set of images focused on the IMC. On the top left, we have H&E. In the top middle, we have segmentation where we identify the single cells. And then, you can, of course, zoom in on different compartments of relevance. On the top right, we've got our major immune cell type. CD30 in green is a tumor cell. And if you can't see very much, it's because there's not very much there. But there's plenty of other immune cells, CD4, CD20, CD68, et cetera. And you can zoom in on different immune compartments, whether it's a myeloid compartment or immune checkpoints, so on and so forth. As we sort of interpret this data, we can identify here, I'm just showing you the 10 major cell types that are present. Includes T cells, the tumor cell is called an HRS cell. And, it's no problem to dig further into immune subtypes and Th1, Th2, M1, M2, whatever you feel like. But I actually don't want to spend too much time on this today. I want to focus on the translation, and I want to focus on biomarkers. So when it comes to biomarkers, the lowest hanging fruit is to just take advantage of all the channels that we have available with IMC. So what I'm showing you here are four different biomarkers that were published sort of painstakingly over the course of many years, right? These are CD68 macrophages, MHC negative tumor cells, PD-L1 positive tumor cells, granzyme B positive CD8 T cells. And these are four different studies, but using IMC, we can kind of in one fell swoop, confirm and validate all these biomarkers as well as proposing new biomarkers, right? So this is nice. IMC, of course, is very good at this because it's multiplex analysis, but I'll say that it's really not taking advantage of the spatial data, right? So I want to move into new biomarkers that really incorporate more spatial analysis. I'll start with just an idea first, though, which is that of ligands and receptors, right? So this is the way that cells often communicate with each other in contact, highly relevant to immunotherapy and immuno-oncology, whether it's PD-1, PD-L1, whether it's the immune synapse itself. But I'd like to offer that, from the perspective of a cell exploring its tumor microenvironment, we should also be thinking about the spatial aggregation of cells, right? And that's a way for a cell to sort of experience an elevated tumor signaling environment, right, through aggregation. And aggregation is, in fact, a very common occurrence in Hodgkin lymphoma. We often see immune cells aggregating around the tumor. They form these structures called rosettes, most commonly with CD4 T cells, but also with other cell types. In this case, I'm showing you CD8 T cells. Sometimes there's very small aggregates like these where tumor cell is sort of touching maybe 1 to 3 CD8 T cells. Sometimes the T cell is sort of completely swarmed the tumor cells and you get a very large rosette. And so we're interested in how the extent of these rosettes affects signaling between these cells, right? So that's what I'm trying to show you here with these plots. Just to walk you through it, starting at the top right, what we're looking at are 6 different colored ridges that correlate with the number of T cells that are surrounding the tumor, the HRS cell, which is the tumor. And so these ridges are colored by the number of T cells, with going from light, which is just one single T cell, to dark, which is over 6 T cells. And the ridge itself is the co-expression of a ligand receptor pair. So in this case, ICOS and ICOS ligand are positively correlated with aggregation. The more CD4 T cells you have around the tumor cell, the more expression of ICOS and ICOS ligand you have, right? And this is not necessarily a given. For example, at the bottom here, we've got HLA-2 and LAG-3. LAG-3 is sort of a next generation immune checkpoint. And we can see that in this case, we're looking at CD8 T cells, but the more CD8 T cells we have, it doesn't affect the co-expression of these two proteins, right? So we have many different cell types available. We've got many different ligand receptor pairs. And what we found is that for every different cell type, as it forms these aggregates, as it forms these rosettes, there's a different pattern of ligand receptor aggregation signal, right? Each cell type has their own unique pattern. This pattern is we of course expect it to be sensitive to patient outcomes and perturbations. And when we look at the relapse samples where these patients have been hit with chemotherapy, we again see this sort of remodeling of these signaling, right? We see less PD-1, PD-L1 signaling, less LAG-3 signaling, and more galectin-9 signaling. So these are sort of, I would say, interesting, curious observations, right? What happens between diagnosis and relapse, but ultimately what we're interested in are predictions. So in this case, what we did is we looked at the diagnostic samples only, and we stratified their patients by the disease severity, right? Between no relapse, late relapse, and early relapse, where early relapse is the most severe outcome. And so I'm just showing you one particular highlight, but we can see that when it comes to PD-1, PD-L1 signaling between CD8 T cells and HRS cells, we only see this aggregation dependent signaling in the most severe cases, right? Only in early relapse. So this is sort of an abstract, right? Like spatially removed observation across many patients, across many thousands and millions of cells. But I think that the key to this observation kind of comes back to this cake, right? Thinking back about these gooey cakes. And again, the thesis here is that I don't actually think that the number of fruits that you put in the cake is the most important thing. I don't even think that the ratio of fruits or the arrangement of fruits is the most important thing, right? There's a lot of ways to make a good or bad cake, but when it comes to cake baking, what makes it good is the bake itself, right? If you have a well-baked cake, you're going to have that caramelization between the fruits, that mixing of flavors, and that's what makes a good cake or not. So here in biology, what we're thinking about is that smudge between cells, that communication between cells as a function of their spatial arrangement. That's what makes a good cake. That's what helps us predict things. Okay, so I talked about spatial changes that we observed after chemotherapy. I talked about spatial patterns that helped us predict early and late relapse. But these are relatively complex spatial features, right? And when it comes to sort of that last step of clinical translation, what we need are simple patterns. This is going back to the very first thing I talked about. So we need simple, incisive measurements that anybody can perform. So, IMC, of course, is a fairly expensive technology. It's fairly laborious, slow. It definitely doesn't fall under the category of anyone can do this, right? But when we're thinking about actionable biomarkers that can be derived with IMC, well, the lowest hanging fruit is, I talked about having all these other biomarkers that we discovered, right? 4 different biomarkers that we sort of instantly validate with IMC. And the lowest hanging fruit is, why can't we just combine these biomarkers into one biomarker? Well, to cut a pretty long story short, that's essentially what we did. So we first derived about 5 spatially resolved biomarkers, right? Looking at distances between cells as a predictor of spatial outcome, as a predictor of clinical outcomes. We use a technique called lasso to select a small number of these that had the most predictive power. And finally, we use a technique called XGBoost to sort of combine these individual biomarkers into a single stratification, right? So we just have high or low based on the score that's derived with this XGBoost strategy. So what are we trying to do here? Well, essentially, we're trying to go from this 35 marker IMC panel and reduce that to a smaller panel of 6 markers of what we think are the sort of 6 most informative proteins that we can get. right? And this in itself is not really is not trivial at all. There's about 1.6 million ways to do that. So there's not enough, samples or time or energy to test all these. But this is what we can do with IMC. And the result is this sort of, reduced protein panel that can be performed with immunofluorescence, multiplex immunofluorescence. The final outcome of this study is actually just distilled into these two survival curves, right? So what are we looking at here? Well, on the left, we have our IMC-derived predictor using these 6 proteins and these spatial metrics. So this in itself, it's a nice classifier, but this in itself is not that interesting if all we're doing is separating these patients, right? You can do this with any data set. You can find some sort of classifier that separates these patients. What's important is that this classifier needs to be validated, right? So that's what's happening on the right side. We've taken a second independent cohort and we've validated the biomarker on this. So this is great. This is important. But I would say that the key of this study and what makes it the most interesting are these acronyms up at the top, right? IMC versus multiplex immunofluorescence. So what happened here? Well, on the left side, we analyzed a large data set. It was frankly very expensive. The machine itself is more than $500,000. But on the right, we validated this technology in multiplex immunofluorescence. Another acronym. These machines are CLIA certified, right? What that allows you to do is actually make, valid, legal, clinical predictions, clinical suggestions using this technology. So what we're doing essentially here, right, is taking something that, again, I haven't actually cooked one of these cakes or baked one of these cakes, so I don't know how hard it is. I'm just assuming that it's pretty hard. But we start from this research scale, very difficult, very expensive mode, and we moved to this clinical scale, right? So Twinkies are sort of our universal symbol of mass production and accessibility. And that's what we're trying to do. That was the essence of this particular study. So again, I won't dive too much into the details of everything, but this particular study was made possible through these 95 longitudinal Hodgkin lymphoma patient samples. The main takeaway here is the gooey cake, right? Thinking about our spatial data, our spatial biology in this sort of, interacting spatial context. And then the last takeaway is just focused on our mission, right? What are we doing with this kind of, multiplexed large scale data analysis? Well, we should be thinking about how to translate our insights, our biomarkers derived from IMC type technologies into something that could actually be put in the clinic via something like multiplex immunofluorescence. So again, if you're interested in either of these two, so this is actually 2 studies. If you're interested in either of them, please check out the QR codes. I skipped over a number of things, including all of our sort of deeper phenotyping analysis, all the spatial clusters and niches, some of the more broad-based biomarker searching. So I'm wrapping up my talk here, but I just want to say, spatial biology, it has a lot of different flavors, a lot of useful applications. So if you're interested in more topics, I hope you'll check out these other papers. We also perform biomarker analysis in another lymphoma type called DLBCL. It's a little bit more clinically relevant, clinically urgent. There's always this question when it comes to spatial biology of what your sort of spatial context is, right? And metastasis is a perfect way to explore this. So we had a study last year looking at colorectal cancer metastasis to the liver. The liver is highly immune privileged, very sort of exotic microenvironment there. We explored that. Along those lines, we just published a study in the British Journal of Hematology looking at, again, DLBCL, but now focused on a different sort of immunocompromised like local microenvironment, right? And in this case, we looked at HIV positive patients by studying a cohort from Malawi, where HIV is endemic. So we can kind of see how HIV influences the local tumor microenvironment and evolution of these cancers. And finally, I just touched on this, but there's this idea of how we can translate this sort of spatial data, how to translate these biomarkers into actionable therapies, right? So this is a preprint that we hope will be out in press soon, where we used machine learning to try and make therapy predictions. So with that, I'll just acknowledge some funding sources, and I'll thank my mentor for the last few years at Cedars-Sinai, Akil Merchant, some of the members of his lab, as well as our collaborators at BC Cancer on the Hodgkins, Christian Steidel, and at UCLA with Sandra Orsulich for the ovarian cancer. And with that, I'll be happy to take any questions. Thank you very much.
Thank you, Alex, for this wonderful talk. So, we will, I think, for now, ask questions. I will get questions. I think Stuni has a question about ovarian cancer is generally cold tumor. Are the immune cells approach can be used to predict recurrence?
Yeah. Ovarian cancer is right. So we'd love for that to be true, but of course, at the sort of I'll just take this opportunity to talk about my thoughts on spatial biology, right? That we've got proteins, we've got RNA. As we evolve, we'll add in more information, whether it's epigenetics or metabolism. But what is protein good for, right? Well, protein has a small number of highly validated targets, right? So I would say each protein has the value of many genes that you might measure with a technique like, this was IMC, RNA, you might use Merfish or Cosmax or Xenium. So I'd say each protein probe has a lot more value individually. So we can look at, actionable targets, right? So what I'm trying to say is that, of course, as we dig into particular immune cells with proteins, you might be able to say more about predicting recurrence, right? Or you might be able to say more about a potential therapy target because it's a particular immune cell type that expresses, like the canonical example is immune checkpoints, right? Whether or not that'll be a useful handle for the tumor. But I will say that hopefully I covered all these different angles in my talk. But if you're just talking about using quantifying immune cells in something like a tissue microarray, there's all these grains of salt that you need to take with it, right? What is the spatial context of the immune cells? Are we sort of being clever about where we're measuring immune cells, or can we believe these correlations that may or may not emerge? So, generally, of course, we think that the more immune cells you have, the better your outlook. But as you dig into it, it really becomes about what type of immune cells are they, what proteins they express. Are these exhausted T cells? Are they central memory? Blah blah blah. Right? There's a the, so immune cells can of course be used to predict recurrence, but we need more context, we need more data, we need more confidence.
Thank you. So I think we have, oh, sorry, we have, I think, another question.
Can I ask a quick question? Because both Yasim and I need to drop out in a minute.
I have also a question.
Okay, go ahead, Yasim. Then I'll.
So, I mean, that's a great talk. I also like your analogy, the cake analogy about the spatial context and how localized spatial pattern predicts the outcome. So my question is, so it may be great for the immune spatial context, but we have a lot of, unknown for the, if there are immune cells are not present or TILs. So would you say that in that context, like if we have like not enough immune phenotype there, using like you showed like receptor ligand together, or RNA velocity would improve the transcriptional states of the cell as a in terms of functionality, or would we be there to use these technologies together to further understand the states of the transcriptional?
Yeah, that's a good question. I'm of course, I think, let's say people like Ahmet and I are probably of a certain generation that I think we're pro everything, right? Like give us more information, more data. If you want to do RNA velocity, that will add context, right? Of course, I don't know. RNA velocity, I think, is a little bit dicier in terms of its value, but if you want to add metabolomics, that's going to teach you more. If you want to add epigenetics, that's going to teach you more. So definitely, it's sort of a very holistic, complex problem, right? The lack of immune cells in itself is an observation. Then what you might do, right? You might have to chase down the tumor, the chemokine production, or you might have to chase down you know, the sort of the adhesion molecules, right? Like, do we have the proper environment for immune infiltration? I mean, on top of all these things, and what's happening more and more in the field is a sort of full holistic whole body comparison, right? Like you can compare your spatial data to something like cell-free DNA or even serum proteomics, right? Like that could add other context for why you don't see immune cells or why the immune cells in the tissue are constantly exhausted. So I I don't think there's everything is a step forward, right? I don't I'm certainly some steps are bigger than others, so there's other some information that you'd like to have first, but it's a complex problem. Right?
Thank you.
So do you think the problem that you had with the ovarian cancer part that you showed were related to sample size?
Yeah. I mean that's really everybody should be thinking about that, right? Like so our sample size was pretty good, right? Like 42 patients we did in triplicate, but there are these cores, right? So the cores are, let's say, a millimeter in diameter, and you're
Well,
always going to have.
Speaking as a clinical person, 42
Yeah.
ovarian cancers is cutting it too short.
Yeah, yeah,
Uh,
that's
and
fair.
particularly if you're using a TMA, I would say that it's really small. And
Yeah.
maybe that's the Achilles heel, so to say, of that particular study. You double the number in Hodgkins and you start seeing uh, quite good results. And it just,
Yeah.
it's related to sample size. And a lot of times when we are talking about these things, we are doing multiple markers on very small numbers and you're eating into your p-value. Very seldom I have seen p-value corrections for multiple analysis. So we have to be careful in designing the study and making sure that you have at least as many samples as markers that you are doing.
Yeah, that's very fair. And the right. So we did, I did report Q values, so we tried to be rigorous about that, which is why there are very few statistical associations at the sort of naive level. You know, there, I would say that the TMA coring, there's a lot of sort of things pulling one way or the other, right? Like any time you work with a TMA, the key phrase is going to be sort of like pathologist curated or pathologist selected, right? And what does that really mean? Well, maybe you're sort of overrepresenting TLS or you're overrepresenting tumor interfaces, right? So everything should be taken with a grain of salt. One thing we tried to do with the digital biopsy is to kind of quantitatively and objectively even the playing field, right? Like if you're looking at a lymph node metastasis, you're going to have all these immune cells that are not necessarily interacting directly with the tumor. So we try to at least address that by sort of shaving off parts of the tumor that may be convolved, right? But this is always the sort of like how much money do you have, right? That's that's the bottom line. How much
Yeah,
money,
but
how
you
much time,
spend
how
all
much
the
energy?
money and don't get any results. That's bad too.
Yeah.
So anyway, I need to drop off.
Thank you, Sunil.
Yeah,
And then
both
we
of
have,
Jessim and I need to drop off. So bye.
sure, sounds good. We have also another question. Tian is asking, what other phenotypes or parameters do you anticipate AI-guided deep phenotyping to identify as biologically relevant?
Yeah. So again, there's kind of two things here, right? Like if there's a sort of discovery-based technologies, which is people generally think about in terms of RNA, and there's sort of more translational focused or hypothesis driven, which is more the protein, right? So if you're using AI tools and on a protein panel, well, you're only going to learn about the proteins that you have, right? So in that case, there's certainly like everything we do there it should be informed by biological ground truths, right? So immune checkpoints are going to be relevant for immunotherapies, different tumor signaling and proliferation signals will be relevant for predicting tumor-based signals. Uh, with RNA and discovery, I mean, this is kind of tying into what Sunil was saying was that uh, you'll see probably everything, right? Like a million things will be AI identified as relevant, and it comes to trying to interpret all that. So I'll say that what we're trying to do, and if you're interested, please check out our preprint. But as far as what we're trying to do with AI and machine learning is, I'll try and distill it here, but the key point is sort of the feasibility of AI predictions, right? Not just predicting any sort of random observation. So our idea was basically to use AI to kind of limit the reasonable parameter space, right? To use AI to say, even if you try to perturb protein X, first you can't like multiply it by 1,000,000, right? That's not feasible. And if the predicted result of your perturbation is to generate, is to increase T cell infiltration by tenfold, well, that's not reasonable either, right? Like that just doesn't happen. So nobody's ever observed that kind of tissue. It's not going to work. So that's kind of, I think, the essence of that particular preprint. So, but that's a good question, right? Like that ultimately everybody is going to have to use AI for this kind of stuff, or miss out potentially, right? So it's a matter of, being, I guess, measured about it, right?
Wonderful. Thank you. Well, I said it's almost 3:05 and we're starting to lose our audience. So I'd like to make sure we thank to you as well. Alex, thank you. I'll be using this clap sign. Congratulations for this fantastic talk. And also, we're glad to see that you are moving to a new job at the University of Maryland. And good luck with the new opportunities there. And yes, I agree with Tian that the good cake analogy, I also love that. It actually made me now more, you know, eat 4 more cakes, right? After spatial. So I think this will be end of this semester, by the way, for the seminar series. And we'll be resuming in August 23rd when the semesters start. And Alex, I think you'll be in Maryland after that. Wonderful. And then hope to see you guys in the fall. And I hope you all enjoy your summertime. Productive, fun, also healthy. And thank you also for your participation. And hope to see you later. Thank you, Alex, again.
Thank you. Bye-bye.
Bye.